Okay. Hello, everyone. My name is Thalia Sass. I'm a PhD candidate in the Lovett Lab. And today I'm going to tell you about my path to science and my current research studying DNA damage and stress responses in E. coli. I grew up in St. Louis, Missouri, and loving St. Louis Blues hockey and still watch most of the games. Specifically, I grew up in a neighborhood called The Loop and around the corner from Washington University. I was fortunate that in my little neighborhood, there are lots of interesting people who had ties to this university. St. Louis is also known as the site for the 1904 World's Fair and Olympics that were largely held in Forest Park. So today, a lot of the museums in the buildings from the World's Fair have been repurposed. The Art Museum and the History Museum, the zoo, all of that is absolutely free for everyone. So my path to science really started by exploring Forest Park as a kid. I found a love of nature in the zoo, curiosities in exploring the science center where I could spend hours on hours in the exhibits in the planetarium. My 5th and 6th grade science teacher, Mr. Byrne, also instilled this excitement for science by keeping the class updated in all the new NASA expositions. One summer, I even went to space camp for a weekend and had the best, nerdiest time of my life. But what it came down to was that I was always encouraged to follow my curiosities. So I went to undergrad at the University of Missouri, about 2 hours from St. Louis. I ended up getting 2 degrees in biology and religious studies and was the first woman of my family to graduate from college. I tried absolutely everything. Took any class that sounded interesting my freshman year, participated in different clubs, joined committees for my sorority and later chemistry fraternity, social justice initiatives. But really, I was finding my passions in what made me happy. I loved spending time at the Hillel Jewish Campus Center, and I loved doing science and research. I was fortunate to fall into the lab of Dr. Samuel Waters my freshman year, and then work with Dr. Chris Lorson the following 3 years studying the disease spinal muscular atrophy. I had no idea I could pursue a career in research, but being around the incredible people in the Lorson lab gave me a framework to see what I could do in science. So all of these opportunities at Mizzou helped me answer these questions. I had a great internship in London at a pharmaceutical company called Pharmadex to see a different side of the scientific industry, and I presented my research at the annual Mizzou Undergrad Research Symposium. I love science and communicating my work to the larger community. So after I graduated from Mizzou and spending some much needed time with my family, I moved to Boston to start my master's of molecular and cell biology at Brandeis, and then I transitioned into the PhD program where I am now. So today I'm going to tell you about the work that I'm conducting in the Lovett Lab. Our lab is interested in understanding DNA replication and repair dynamics in bacteria, specifically in E. coli. So E. coli grow and divide really quickly, especially when there is plenty of nutrients and the conditions are just right. We can plot E. coli growth by measuring the density of the culture. When the media is getting really crowded and the nutrients are running low, the cells need to adapt to slow their growth with this new stress. The cell changes the genes that it's expressing, and that changes their ability to respond in this dynamic environment. Part of the central dogma, DNA codes for RNA, codes for proteins. The machine that's reading the DNA is called RNA polymerase. And there are adapters on RNA polymerase called sigma factors that can change what part of the DNA is read to produce RNA. So when the cell is transitioning from the fast-growing log state to the hunker down stationary phase, sigma factor S modulates this change and regulates about 10% of the E. coli's genome. The cell has a whole network of regulations, and we use basic techniques to figure them out. For instance, we know that RPOS is typically degraded by a protein called RSSB when the cell is growing really fast. But when there is a time of stress, RSSB is inhibited by anti-adapters, one being iraD. iraD prevents sigma S's degradation so it can interact with RNA polymerase and initiate the general stress response. So how does the cell sense stress, specifically DNA damage? We have to work backward in our network, and thus we have to understand how iraD is regulated. We can measure when iraD is being turned on in cells by using light. What we do is we put the gene's promoter in front of a protein called luciferase that was actually discovered in fireflies. That's what makes them glow in the dark. Our machine can measure the light from this gene, and we can divide it by the amount of cells in the culture, and that gives us the relative luminescence units, and we can plot this on a graph. Here is the iraD relative luminescence units plotted over time. We treated the lighter points with our DNA damaging agent, AZT, and we can see induction right after that treatment. We can also see induction later when the cells are entering stationary phase and feeling the stress of the nutrient starvation. We can then form a hypothesis about how iraD is regulated based on what we know from previous research. So we know it's induced with DNA damage, it's inhibited with rapid growth, and it's induced with the starvation hormone called PPGPP. And this led us to test the hypothesis that the protein SSPA, or stringent starvation protein, could be having an effect. So we created a mutant in E. coli for SSPA and measured the construct in this cell line, seen here in blue. We see that SSPA both has a higher induction after DNA damage and an overall lower baseline level, and we found this dual regulation really interesting. We also know that iraD has a really long upstream region with 2 different promoters. So we mutated one promoter or the other to see if they have a specific roles in this dual regulation. So the SSPA phenotype is most apparent in P2 that responds directly to the DNA damage, but not during starvation. P1, on the other hand, turns on when the cells are feeling that nutrient stress and mildly with DNA damage too. So what I've shown you here is this link between SSPA and iraD, but these networks that we can create with gene regulations are ever growing and we're always finding new answers, but we also have so many more questions. And E. coli are really great model organisms for looking at these answers because they have a small circular genome and all of the code that they need to live and survive in these harsh environments are encoded in these 4.6 million base pairs that's all crumpled up in a circle stored inside the cell. So I'd like to thank, first and foremost, Waltham High School for inviting me here in my lab that's led by Dr. Susan Lovett and everyone else in the Lovett Lab who has contributed to this research, the Science Communication Lab and Anahita Zaire, who's the director, Brandeis University in the Molecular Cell Biology program, and my funding.